So basically, I'm going to talk about neural communication and the parts of the cell. And so basically, this is a cell, this whole thing right here, that is labeled with the different parts and the functions. So you have your cell body, which is the cell's life support center, and it has the nucleus and things like that. Then you have some dendrites attached to the cell body, which receive messages from other cells and say it back to the cell body, so to transmit the messages. Then you have the axon, which it passes the message away from the cell's body to other neurons and glands. And then you have the nodes. Then you have the myelin sheath. Basically, it's a fat that kind of covers the axon, and it helps transmit the messages that goes down the axon, which are electrical messages, and it speeds up the neural impulses. And then these little creases right there within the myelin sheath are called the nodes, and they help have the transmessage jump. Then you have your terminal branches, which form junctions with other cells. And then you have your synapses, which are contact points between neurons, and they almost touch the neighboring axon, but they don't, and so that there's a little gap between them, and that is called the synaptic gap. Then, you have neural impulses, but that's basically, we're going to talk later in neural communication. So the first step into the neural communication, there are 6 steps. You have your resting potential. So this is the cell body, this is the neuron, and basically you have potassium on the inside that's negatively charged, and you have sodium on the outside that's positively charged, and then you have your sodium potassium pump right there. And then basically resting potential is when this inside is -7 millivolts, and the sodium potassium pump creates a resting potential and basically acts like a gate. The dendrites, which act as sites that receive signals from other neurons, get the message from the other cells. And then your next step is going to be depolarization. So this pump you're gonna have sodium rush into the cell body as potassium rushes out, and this is happening all through the sodium potassium pump. And then the dendrites go into the cell body next. Then the third step you're going to have is a threshold. So basically, the threshold is when enough sodium ions rush into the axon hillock. And so when the sodium is able to rush the cell body through the voltage-gated channels, and the potassium leaves the cell body, not causes, and the potassium is going to leave the cell body. And yeah, then your fourth step after is called action potential. So basically, action potential is it's going to be a message that's sent away. So the positive ions go in guidedly quickly to create the desired domino effect, and the voltage-gated ion channels that are on the axon open when the sodium is going to rush in. And the whole process gets sped up by the myelin sheath, and makes it more efficient. And also is helped out by the nodes of Ranvier. And yeah, then after that, you're gonna back to this whole process of action potential. It's called salutary conduction. Not the process, but the process of this with using the myelin sheath and the nodes to speed up the process is called salutary conduction. And basically what that is when the signal jumps from one node to another. Then next you're going to have a refractory period, which is not shown here, but there is one here. So basically a refractory period helps direct action potential down the axon, because only channels downstream can open. And the neuron becomes polarized. Sodium potassium pump makes sodium ions go outside the neuron. And then after that, you have your exocytosis. And basically, the neurotransmitters need to get released across the synaptic gap. This occurs because action potential This occurs because of the action potential, and the electric message goes to the terminal branches. It shocks the vesicles holding the neurotransmitters. The vesicles moves the membrane attached to the cell membrane of the terminal branches. Some neurotransmitters that go into the synaptic gap attach to the ligand-gated channel in the dendrites of the cell. And that you could see that there. Then you have your reuptake, which is the last thing, which is basically the neurotransmitters get recycled by the uptake parts. And you have your serotonin receptors and your vesicles that are housing the serotonin that's already up there, and you have your blockers. And we do this because basically neurotransmitters are really expensive to make, and so we just recycle them. And so yeah, that is the process of neural communication and some parts of the neurons. And there are some notes for more detail. And yeah.